Nigel talked about the tongue and papilla in lecture last week, but we held the slides off this week to distribute not necessarily number, but information content. So what kind of papillae are these? Filiform papillae. In humans, they're developed like that. As Nigel mentioned, if you've ever had your cat, your domestic cat, lick you, you can see that they're really carotid. Imagine having a big felid like a tiger or lion lick you with filiform papilla, and it can literally lick the skin off your body, which it has. Humans that have been unlucky enough to be preyed upon, or lick fur or feathers off their prey. All right. So evolved filiform papillae are good things for big predators like cats. So we've got the epidermis here. We've got a little bit of lamina propria, and we've got striated muscle. What are these spaces here?
Salivary.
Cross-section of this connective tissue sorts of thing. Here we've got a circumvallate papilla. Remember, these are found in the posterior aspect of your tongue, in the terminal sulcus. The whole papilla is this whole structure, including the trench that encircles it. You can see nicely the taste buds here. Here is a duct. What kind of gland is this? Serous, right. Von Eppner's gland, if you want to get fancy. But just by staining characteristics, it's not a mucus gland. Although there are mucus glands in this vicinity. Here is a higher power shot of that previous trend. Oh, you got a mitotic division right there. But you can see these receptors, gustatory receptors, have an actual sensory cell, a supporting cell, and a basal cell. Much like the olfactory receptors, the olfactory epithelium, only they're clustered in a little globule. You might almost be able to hallucinate the pore through which the microvilli that bear the receptors for the taste would hang out in here. Von Eppner's gland is clearly going to provide a serous solution that these molecules can be dissolved in and then stimulate your sense of taste. Does anyone, what are the modalities of taste?
Sour. Acid.
All right, I heard 4. Where's your 5th?
Umami.
Yes. Umami. That's for fats, amino acids. It's recently discovered. U-M-A-M-I. Umami. I think it would be a Japanese. Right, from Japanese word. Okay, low power shot of the parotid. Why is it the parotid? Well, from what you can see, all serous. Why is it parathyroid? What do we see here that would eliminate the parathyroid right off? Yes, ducts. Parathyroid is an endocrine gland, lacks ducts. So serous acini, we're going to ignore adipocytes as a discriminating characteristic. Higher power, showing the serous acini. Here you can see what looks like a striated duct. You can almost hallucinate the striations there in the basal aspect of this cell here. It's a transporting epithelium. It modifies the electrolyte content of the saliva. High power shot. Don't have a low power context, but you should be able to tell me that this is the right. You've got mucus and seromucus acini. You've got the serous demilins. You can even see the enzymatic granules in the cytoplasm, in the cytoplasm of the cell. What is this? Yeah, it's a duct. What kind of epithelium is lining that duct? Probably, I would argue, columnar. I mean, this cell is a little wider than that cell, but clearly stratified columnar. It tells you this is a larger excretory duct. That happens to come from the carotid. In connective tissue, what are these? Not an exam, so sing it out. No prize for giving the wrong answer. Nerves, exactly. That's the autonomic innervation that would run in the connective tissue space until it gets out and ramifies into the parenchyma of the brain. Bless you. We're not expecting that you identify this with glass slides, but for completeness, what is being produced here? This is the lingual, sublingual. So yes, lots of... okay, low power shot. What tissue? Right, and what are your landmarks? Exactly. And this one, even at this low magnification, you compare, look, we've caught section through the wall of the bile duct here. Here we've caught the lumen. You pick that up, it's clearly a cuboidal epithelium. That little scrunch down area, probably hepatic artery or branch thereof. And right here, most likely, portal vein. This could be lymphatic space. Look, I've got a higher power, which shows you what you could already pick up at low power. And that's, once you get the search image in your mind, you don't need to get up to high power, you get a feel for it. If we back off here, so this is probably central vein, or more properly, terminal hepatic venule, which will coalesce into a central vein. So which way is blood flowing? This way? Right. And you've got bile and lymph flow here, which makes perfect sense because you got a duct here. Remember that the plumbing, if you will, that the bile goes throughout here are those canaliculi, which are small little chambers carved out between two hepatocytes linked together by tight junctions. So bile and lymph flow is that way. And you can see, even at low power, what we mean by cords in two-dimensional, or in reality, are plates of hepatocytes. I mean, you can just follow them, interconnected cords, strands, separated by these clear spaces, which are the sinusoid. Yes. And this is exquisite shot of a bile duct there. All right, so we can see the hepatocytes, we can see the sinusoids, and note that these Kupffer cells, which are macrophages, which have ingested the carbon particles from Indian ink, which was injected into the animal's vascular space before it died, is accumulating in the Kupffer cells, and for the most part, they're hanging out in sinusoidal space. That's not to say that they don't take a process every once in a while and probe the space of Dis, the parasinusoidal space of Dis. But for the most part, they're hanging out in sinusoidal space. I showed you this micrograph in lecture. So the sinusoid is kind of bending this way.